In this chapter, let's break down the science using 3D visuals to show how the mRNA vaccine works and explore its potential links to clotting and heart damage. But first, let's recognize that messenger RNA isn't just a man-made invention. In its natural form, it's a marvel within our bodies. Evolution has gifted life with the wonders of genetic coding, the blueprint of existence. In this grand symphony of life, messenger mRNA plays a vital part. The magnificent diversity of life stems from humble beginnings, evolving over billions of years to attain its marvelous complexity. The journey from primitive beings to complex creatures would not have been possible without the genetic blueprint called DNA and the related molecule RNA. DNA holds all the instructions to maintain the body. These instructions are then copied from DNA into smaller snippets called messenger RNA, which are the to-do list for each cell. So you can think of DNA as the blueprint, and mRNA is the project manager, bringing plans to life.
We choose to go to the moon in this decade.
During the space race of the 1950s and 60s, genetics research saw rapid progress, including the discovery of messenger RNA in 1961. Today, scientists have figured out how to create a synthetic, modified version of mRNA in the lab that is engineered to last longer and evade immune detection. Our cells mistake this lab-made mRNA as their own, and our body starts to create proteins for therapeutics or vaccines. The modified mRNA vaccines are created on a computer in a lab, without using a real virus. These genetic vaccines only required the genetic sequence of the coronavirus.
We never had access to the physical virus to design the product.
In the early stages of the pandemic, Chinese scientists released the genetic code of the coronavirus, sparking a global race to develop a vaccine.
As soon as we get that sequence, let's plug it into mRNA. That's all I needed was the sequence.
They used the genetic code provided by Chinese scientists as a backbone for their mRNA vaccine. The modified genetic code in the vaccine instructs our bodies to produce only a specific part of the coronavirus rather than the entire virus itself.
Vaccine platforms are using what's called the spike protein of the coronavirus.
The spike proteins are the tiny spikes on the surface of the virus. Once this mRNA code is injected into your body, it instructs your cells to make these coronavirus spikes. By creating these spikes in our body, our immune system should build immunity against this specific part of the virus.
The worst mistake of all would be to take messenger RNA and then have it code for a lethal protein that was engineered in a Chinese biosecurity lab. That was a disaster.
The mRNA technology is exceptionally complicated and unlike anything we have seen with standard vaccines.
When people received their first COVID-19 vaccine, it was the first time their body received lipid nanoparticles.
The mRNA vaccine dose is filled with tiny bubbles of fat called lipid nanoparticles. These fat bubbles protect the modified mRNA from degrading and help it enter cells. Once inside, the mRNA instructs the cells to produce a genetically engineered version of the spike protein.
The lipid nanoparticles, when they attach to cells, then the lipid nanoparticles dump off their payload, messenger RNA. The messenger RNA then is taken into the cytoplasm of the cell. It's read by ribosomes over and over and over again. The spike protein, which is a foreign protein, is expressed on the surface of cells.
The injection of mRNA vaccines turns our bodies into spike protein manufacturing sites. With every dose, a substantial amount of these modified spike proteins are created inside your body. The scientific community has stated that the spike protein created by the vaccine is harmless. Concerns have been raised about its potential to cause harm. A Yale preprint detected spike protein up to 709 days after vaccination in patients with post-vaccination syndrome.
The code that they used is for the spike protein. Early on in the pandemic, we learned that the spike protein is the toxic part of this virus that causes clotting, that causes inflammation, that causes myocarditis, that causes brain fog.
The spike protein directly causes blood clotting and is found in the middle of large blood clots.
Researchers have concerns that the immune response triggered by mRNA vaccines could potentially lead to excessive attacks on our tissues and organs.
And any cell that makes this foreign protein now becomes a target of your own immune cells to attack as well.
Those cells will be attacked immunologically, as if they're infected with the virus.
The immune system detects the presence of spikes and initiates the destruction of the infected cells. These cells become a target for our immune system to hunt and kill.
It immediately evokes an autoimmune reaction in each and every cell that is expressed.
Health officials have been cautious in acknowledging that these particles may have the potential to distribute beyond the injection site. If these particles enter the bloodstream, they may potentially increase risks. A widespread distribution could result in the production of spike protein in various organs.
Lipid nanoparticles go everywhere in the body, as in everywhere. To the brain, to the bone marrow, to the liver, to the spleen, most importantly to the reproductive organs, to the testes, to the ovaries. So lipid nanoparticles can't be controlled.
Most of the lipids should stay in the shoulder, and they are designed to efficiently enter the deltoid muscle cells, but they can also be taken up by other cells in the body. So not only can the cells in your deltoid muscle be turned into spike production sites, but also cells in your heart muscle and organs. Current leading theories of mRNA vaccine-related cardiovascular problems include spike protein expression in tissues and molecular mimicry of the spike protein. The late pathologist Arne Burkhardt and his team examined over 100 biopsies from individuals suspected of having suffered adverse reactions to vaccines, some of which were fatal.
And the destruction of the endothelium may be one of the major factors.
These biopsies provide clues about how the vaccine may contribute to damage to organs and tissues.
Our findings are so alarming that the vaccination should be stopped immediately, and especially the vaccination of children.
When these tiny fat particles enter the bloodstream, they may be absorbed by the walls of blood vessels or the heart. If these tissues produce spike proteins, our immune system may attack these areas, causing potentially serious damage to the vessel walls or the heart.
We found spike protein in many organs and many tissues. Spike protein was in the endothelium and in the vessels, in the vessel walls.
Arne Burkhardt's autopsy findings align with a growing body of biopsy and biodistribution studies, including one that used deep learning to track lipid nanoparticle distribution in animals.
It was actually known before it was deployed in humans, based on rodent models, that these products went all over the body, and yet they persisted to continue to deploy them.
Health agencies approved mRNA vaccines, knowing that lipid nanoparticles can spread to different organs in animal studies.